Today, chemical library space. I have some 30 slides or so. Okay, right, it looks like it's working. So I will introduce a little bit chemography as a principle, and then our favorite chemography tool with this generative topographic mapping. And then we come to the actual topic, chemical library space, by contrast to chemical molecular space, which is the classical standard of chemography, and we'll show you how we intend to use these tools to allow the end user to switch paradigms and manage a portfolio of libraries in a way in which one can manage one library by modulating the content in terms of molecules. So let's start with the beginnings. You know, chemography is it's a rather fruitful idea by a certain Tudor Oprea, which at that time worked for AstraZeneca in Sweden, who said, well, we all know that molecules can be characterized by many different molecular descriptors, which basically define a high-dimensional vector in some abstract space, and intuitively contemplating high-dimensional spaces typically results in a headache. So what if we force this space, we flatten it down to 2D? Technically, that means we use a dimensionality reduction tool. Mathematicians are very good at this kind of tricks, and there are many ways in which one can flatten down an n-dimensional space in a way that preserves the neighborhood. So typically what you want to see is that items that are close in the high-dimensional space remain close after being projected on the 2D map. I'm not going to talk about all these approaches. Here you see that most of these approaches are able to comply with neighborhood behavior, in the sense that here we represented from the Dude database, we have taken 100 actives, 100 inactives, acetylcholine esterase inhibitors, and as expected, they tend to cluster together in the sense that the close contacts between blue dots and red dots are, as far as possible, minimal. Well, I'm not going to go through all this. We will just focus here on the lower corner with this very colorful landscape, which sets the tone. That's why we like GTM, because they're colorful, okay? And maybe not only that way. So technically, GTM works as following. One has a chemical library, well, a chemical problem, like we want to map, right, a representative chemical subspace. Let's be modest, we're not going to ever map the entire chemical space. We just focus on some, like drug-like compounds or fragment-like compounds, or PPI, protein-protein interface like compounds, and so on, whatever. Then we need to fix this subspace by selecting a representative frame set. The frame set is here just to fix the space. It's like the points that are chosen by the geometer to install the theodolite or whatever instruments they use to build a map. And you don't need to do the measurement in front of the Sydney Opera in order to get a map on which the Sydney Opera can be very well located. So the molecules that serve for frame set are not necessarily the bioactive molecules we want to analyze, but they should be, let's say, they should populate the same chemical space area, because if you want if you establish a map of Sydney, you will have a big problem to place the Liberty statue from New York in there. So it's just good old common sense. So these many molecules are represented in their high-dimensional descriptor space, and then the algorithm inserts a two-dimensional flexible manifold. It's like a rubber sheet, and it's it makes you think about general relativity, which is the manifold where space-time is the manifold that is curved by mass, while manifold is curved by these molecules, and its shape is optimized in order to pass closely to as many reference frame set molecules as possible. The shape is rendered by the use of some Gaussian functions. Mathematics in there is not particularly intuitive, but the end user couldn't care less. So the manifold is fitted. This is an optimization process. And at the end of the day, the manifold is saved and is flattened out and is represented as a 2D grid of points. And the real game changer in all this is the fuzzy way in which the items are then eventually mapped. For those of you who are familiar with Kohonen maps, you know that at the end of the day, one item goes to the closest node and it becomes a 100% resident of the closest node. Well, in GTM, this is not the case. One item will be fuzzily spread, will have multiple part-time residences within all the surrounding nodes. And you will have the molecule will be represented on the map by a cloud of, you could call them probabilities, but I don't like it because there's nothing stochastic in here. This is just degrees of association, and the technical term is responsibility. So this is a responsibility vector that defines the mapping of my compound. If you really want to have the compound in one point on the map, it will be the mass center of the responsibility cloud. But beware, right? You know, this is like if you have a guy who lives part-time in New York and part-time in Paris, this cross will be in the middle of the Atlantic. It's not a good choice for the residents, but beware of artifact if you really want to go to this point representation of items. It's possible, but not recommended. Stick to the responsibility cloud. It's a much richer way to look at the items, and sometimes it can be that the cloud collapses to one node. So occasionally a GTM may act like a standard Kohonen map. Okay, and so why is this interesting? Well, you can actually do some rounding up of these real number vectors. You say this guy is a 3 per mil in node 12, 51% and 48% in node 37, and then some residual in node 77. Ignore the residuals and round up these two, and so this will give you a kind of a street address label. And why is this interesting? Because it's like a cell-based clustering method. It's very fast. You can quickly group molecules that map together by this responsibility pattern and list them out. And of course, when you can map one compound, you can map multiple compounds. You see there's a density landscape building up, like a cumulated responsibility landscape. And then the idea is straightforward. This kind of strange drawing, this kind of Rorschach pattern is somehow characterizing the library that you are being mapping. So why is this relevant? Well, this is relevant if and only if the GTM is neighborhood behavior compliant. But if the GTM is well built, then of course it will tend to regroup molecules that share some common chemotype together on the same map zone in the same responsibility pattern. What is this chemotype? So this is the beautiful part of it. It's not fixed. It's not, should I say, predefined. It is just learned during the mapping procedure. Sometimes a chemotype can match a given scaffold. Sometimes it is a mixture of very relevant scaffold or very similar scaffolds. Sorry. So imagine heterocycles where the nitrogen atom moves a little bit to the right, to the left, but finally it's the same class of compounds. Sometimes it can be a same topological pharmacophore, like Zwitter ions will typically cluster together. Sometimes it's just, in particular in the corner, it's a little bit like the famous garbage nodes of the Kohonen map. You will have all the very small molecules or the very lipophilic molecules clustering together. But globally, we showed this kind of clustering to medicinal chemists, and they typically agreed that it makes sense. So insofar, this is looking at these responsibility landscapes is supposed to make chemical sense. And furthermore, if you start coloring the landscape by the nature of the resident items, you can obtain various predictive landscapes. So, for example, you can compare two libraries. If you color the members of one library in red and the others in black, and then completely black, completely red nodes are exclusively populated by the members of the two libraries. Nodes in which the two populations mingled have intermediaries. So you have a very visual view of the overlap. If your two categories, library 1 and library 2, represent the actives versus the inactives to a given target, visualizable QSAR model, because you know that a new molecule projected in red will be likely inactive, and projected in black will be likely active. See what I mean? So we have density landscapes, class landscapes. The third would be property landscapes, where you color by a continuous property such as log P or molecular mass or PIC50, you name it. Why? So GTM nodes act as knowledge repositories. Take a molecule, this molecule is active, it has a PIC50 of 7.8. So what I do when I create the landscape, I will increment the node PIC50 by 7.8 multiplied by the residence time. So the nodes in which this molecule resides a lot will be impacted a lot by this 7.8. The others will be less. And at the end of the day, after I project all the known molecules with measured PIC50s, I just need to normalize the node values, and I obtain the specific node PIC50s, which then can serve to predict the newcomers, right? So furthermore, the beauty of GTM is that this model comes with its own applicability domain. If you say, okay, a new molecule, a candidate to be predicted, is projected onto a node that is hardly populated, well, you can extract an average PIC50 for that one, but I wouldn't trust it. If I have a node in which neighborhood behavior is not clear, in the sense that it has residents with both high and low PIC50s, I still can calculate an average, but I wouldn't trust it. So we can define a trustworthiness score and look at it, look at predictions from this perspective. We're not going to go into that kind of details today. Just keep in mind that GTMs are like a Swiss knife. You see, we started discussing chemography. We finished discussing predictive models. Well, with GTM, you get it all in one, and you can actually do obvious predictions. As I said, if this guy falls in the white zone, it's out of applicability domain, and so on. Okay, well, there was an if, which I mentioned before, if the map is neighborhood behavior compliant. So let's make sure it is. And to this purpose, we built what we call universal maps. There's a lot of degrees of freedom when you build a map like this. First of all, it has the intrinsic parameters, which I mentioned, the number of nodes on the grid, the number of Gaussians, which are used to model the manifold. Like the more Gaussians you have, the more flexible the manifold becomes. And foremost, the big question is, okay, I'm doing dimensionality reduction of a descriptor space, but what descriptor space? Not all the descriptor spaces are equally suited for predictions of a given property. So we left this question open, and we considered the descriptor type as a degree of freedom of this optimization. So we used fragment descriptors. Our ISIDA fragmentation tool supports many possible fragmentation schemes. It basically can emulate all the Morgan and ECFPs and even other more exotic fragmentation schemes. And what is the fitness function of our map? Well, it's polypharmacological activity prediction propensity, in the sense that we didn't want a map that works well for mu opiates or for COX-2 or for adrenergic receptors. We wanted a map that works well for as many targets as we can get. So we fished out more than 200 structure activity sets from Campbell, and we asked the map to, we asked the genetic algorithm to find us a map that works as well as possible for each of these individual prediction problems. Because this is the beauty. If you have one manifold, you can use it to predict many properties. It's just a matter of coloring the manifold by the training set of the given property and then reading off newcomers from the landscape like this. So this is the same manifold, it's just the sets that are projected in there are different. And of course, like with any QSAR, there are some targets that have a better balanced accuracy in terms of, this is cross-validated balanced accuracy. Trypsin inhibitors tend to be very diverse, and it's difficult to separate them. By contrast to dopamine inhibitors, where there are clear clusters of actives rather well separated from the rest. Mu opioids is the same, but we have one map, many models, which makes GTM actually a very good pharmacological profiling tool. And we use several maps. These are based on different molecular descriptors. Some are better descriptors of the molecular topology. Some are better descriptors of the molecular topological pharmacophore. And each of the maps is allowed to make a prediction. And then some maps will say, I don't know to predict this guy. And then there's a consensus prediction that gives actually rather robust outcomes for more than 700 targets now, because we use 230 to fit. And then we could show that the predictive power extends to many other targets, up to 750 that were validated in this way from Campbell. Okay, so I hope I convinced you that generative topographic mapping is something worth looking into. And now let's come to the main topic, describing a chemical library as a standalone object. Well, why is this? Well, for obvious reasons. In a screening library, which comes in wells, right, you could, in principle, do cherry picking and make your screening campaign in terms of selecting individual molecules, but sometimes you simply wish to look at the library as a whole. In particular, when you do library comparison, it makes sense to characterize well, the corporate library versus the Sigma library versus the enamine library, and so on and so forth. Well, there's another beast on the market, the DNA encoded libraries, the DELs, which are mixture combinatorial chemistry. And in this case, the separation of the individual molecules is not an option. So we should characterize a DEL as one indivisible entity. And as I already hinted, one good way to do it, the way we suggest, is to use these density landscapes, which are nothing else but a vector, because you just have a density landscape is what? Is a set of values specified for each of the nodes of the GTM. They're accumulated over the library. And then you can have either density, the way I already showed, or relative density if you want to make it library size independent. Otherwise, of course, the higher the library, the higher the cumulative responsibility values. This makes sense. You could have this landscape modulated by a reference library. For example, I want to give more weights to the nodes that are seen to be populated by a drug-like library, or by a library that is tuned for my receptor, or for some library I prefer. So I stop giving equal weights to nodes. I give higher weights to nodes that are reputedly populated by molecules I kind of care for. Or I can weight these nodes by property. So distinguish nodes from the hydrophilic regions, from nodes from the hydrophobic regions. But nonetheless, this landscape is a vector, and the vector describes the library. We saw that the nodes that define these vectors can be empirically associated to some chemically meaningful chemotypes, whatever they are. And well, then life is simple, because if you ask me the question, how do you calculate the overlap between two libraries? Well, it's just a matter of covariance of their characteristic vectors, right? Here we compare Campbell to zinc real, Campbell to zinc tangible, in the lead-like chemical subspace, in the drug-like chemical subspace. You can play around with these vectors, define them in a way that suits your problem, and then use the Tanimoto score in a good old way. Knowing that whatever the Tanimoto score returns to you, it's a value that is always easy to grasp, because you can always fall back to the landscape. Say, okay, wow, look, this is a nice match because we have a lot of green. Green means that these are the zones that are rather equally populated by the two libraries. With the zinc tangible, there's a zone that is rather specific to zinc tangible. It's not so well populated in Campbell, and so on and so forth. So, of course, you can formalize these and define coverage according to various metrics. You can look at shared responsibility patterns. You can look at Tanimoto coefficients, as I already hinted. And each of these typical these characteristic vectors give you a specific response. When you look at cumulative responsibilities, you just assess how much do the libraries overlap. So how much of the chemical space is common to them. If you look at property modulated vectors, it will tell you how much they overlap, but within the log P zone between -1 and 2, if that's what you're looking for. If you look at the library class modulated vectors, it will tell you how much they overlap in particular for those nodes that are also populated by a reference background library for which I care. Okay, and the interesting thing is that the CLS patterns, they encode the chemical composition as property distribution alone, which is the standard way to look at libraries, does not. So here you have 3 density landscapes. One is Campbell and 2 DNA encoded libraries. If you look only at the log P distributions, you would say, well, what matters, del 2568 or del 271? It's the same log P distribution. It matches equally well or equally badly the Campbell log P distribution, so why should I care? That if you look at the chemical space occupation, there's an obvious reason to prefer the first del over the second del, if of course what you're looking for is a general purpose del for primary screening. If on the contrary you know that your target you're hunting for prefers to have actives in this zone, then you will prefer the second del because it's smaller and more convenient. So it's just plain common sense. And finally, so this, now we have the tools. Let's see how to play around with them, right? To manage a portfolio of libraries. The paper just came out, and so an exercise we did is given a set of dels. So it's like we used Eli Lilly tools with associated building blocks, and came to the conclusion that there would be some 2,500 different dels that could be synthesized in principle and enumerated. The question was, which of these dels are the best replacement for the Campbell collection? So we just explicitated the products in those dels, not all, just enough to obtain some representative density landscapes, and then we ranked these dels by overlap to the Campbell density landscape. And of course, you can do it as I told you. There's various metrics here. There's various vectors. This is responsibility pattern based, with or without weighting. This is background library weighted landscapes. This is normalized cumulative responsibility. I mean, the ranking is done according to these metrics, but what is depicted is the density landscape, and you see that depending on what metric you use, you don't have the same del that wins. So each metric will give a different winner, a different del in 50th position, 100th position, and so on. But no matter what metric you use, you observe that the main contribution to these scores is the overlap, the commonly shared chemical space. So all the first ranked dels have a significant degree of overlap with the Campbell landscape. The latest of the dels are rather small and they don't overlap a lot. And then it's up to you, depending on your project, to decide which scenario is the best for you and pick the corresponding matching del. So you can already see trends here. Although the best matching del might depend on the metric you used, you see that they all have something in common. They are all dels based on robust coupling reactions. So they are 3-step dels. This one requires 2 steps of reductive amination followed by an Ullmann, and so on. They are all based on good old workhorse reactions that are much loved by the chemists, and it's not astonishing that these reactions have a lot of building blocks at hand. There will be large DELs that are obtained, and they will cover the chemical space of DEL better than the very special DELs that are obtained with rather exquisite heterocyclization processes, for which the building block diversity is rather low. So at this point, we could think about these DELs as items in a chemical library space. And there's the multidimensional descriptor that is obtained from the mapping landscapes of these DELs. What if we take this descriptor and throw it onto a Meta-GTM? Why Meta-GTM? Because they are descriptors already produced by mapping, and now I am building another map that dimensionally crushes the mapping descriptors from step one. And in this Meta-GTM, libraries will project in the same way as molecules projected on a library landscape. Right? So we will have specific positions for each one of the DELs and Campbell and zinc, whatever we want to project in there. But does this make sense? Well, if you look at it, if you consider the two classes, we have taken the DELs in the spirit I've shown before. We've taken the ones that are the top 100 overlappers and the rest of the world. We defined two arbitrary classes, well, not arbitrary, but two classes, and we looked at the Meta-GTM landscape colored by this classification. And the good news is that indeed the zone closest to Campbell maps here into this corner because it's an atypical library. This is a DEL landscape. Campbell is a bit of an outlier. If you project it, it will project somewhere, but it's squeezed a bit in the corner. Nonetheless, the nearest neighbors, they're not very near. You saw that there's no single DEL that perfectly matches the Campbell landscape, but the nearest neighbors continue to be the closest one after the projection. So this is start coloring by other properties. For example, we could classify the DELs as a function of the number of steps that are involved. There are 3 building block DELs and there are 2 building block DELs. And well, there's no real trend here. The library space doesn't seem to be split according to the number of building blocks, which actually makes sense because the number of building blocks is a relative issue. It depends on how you define building blocks. After all, you can take 2 building blocks, couple them together separately, and make another building block out of that will couple with a third partner, but it will count as a 2BB DEL. So this is completely understandable. However, if you start coloring by the number of heterocyclization processes involved in the synthesis, then you start seeing a pattern. The DELs without, so the DELs without heterocyclization, the DELs based on standard good old coupling reactions, occupy all this chemical space closest to Campbell, and in let's say northeast, we have the heterocyclization based DELs, which actually go scout some new chemical space. This is an interesting observation. If you look at purely heterocyclization based DELs, so where all the reactions involving are heterocyclizations, you find a very restricted small family, meaning that, well, they're not so popular yet. There's something to be explored in this area. this domain is not fully visited yet. So that brings me to the conclusions. So with a well-trained map, and we can check whether a map is well trained because it can serve as a predictive model, and if it is a successful predictive model, implicitly means it is a good map. So the cumulative responsibilities will then allow to describe libraries in terms of chemotypes and are therefore very good library descriptors. That makes libraries become standalone objects like molecules in classical chemography. You don't have one molecule, one vector, but now you have one library, one vector, and that makes life really easy. Finally, we saw that these vectors can be modulated by compound properties, so you can play around a little bit in response of the peculiar needs of every project. And last but not least, this vector space can itself be subjected to mapping in a meta map where libraries will be depicted by their meta responsibility patterns, yielding analyzable maps that can give an overview to the managers in screening projects. There are some articles available online. You scan these QR codes quickly, or else I don't know if the slides will be available to you after the presentation. If you don't get to scan them now, just contact me. And I wish to thank all my colleagues who basically contributed very much lots. In particular, Juliana, who's now with Eli Lilly in Madrid, and one of the twins, don't ask me which, I can't discriminate them, they're too close on the GTM. Alexander Varnek, you might know, the head of the group, Jill Marcoux, my colleague, and everybody else for the excellent atmosphere here at the Chemoinformatics Laboratory in Strasbourg. And herewith, I wish to thank for listening and waiting for your questions.